In this video, we are going to be covering the topics in Chapter 18, which include storage mechanism and control in carbohydrate metabolism. So in this section, we are going to be learning about how glucose molecules are stored in the body and how this the biomolecule that we use for storage is then utilized in order to produce glucose under various cellular or environmental conditions. So, understand that when we digest a meal that is high in glucose, that excess glucose needs to be stored. The two organs that we see the storage of glucose are liver and muscles. When glucose is stored, it's actually stored in the form of glycogen. And the molecule that we have here to the right of the slide is actually the structure of glycogen. Glycogen is also known sometimes as animal starch because remember that when it comes to starch, starch is made out of amylose and amylopectin, but when it comes to which one of the two is glycogen closer related, amylopectin will be the one because remember that amylopectin is branched. The metabolism of glycogen is specifically what gives us an insight into how glucose is made on demand when the cell needs it. So, as I just mentioned, when it comes to the storage of glycogen, as you can see, we're going to be observing this storage specifically in liver and in skeletal muscle. Specifically, the enzymes that are involved in the cycling of glycogen, meaning the breakdown but also the synthesis, are going to be the following. Glycogen phosphorylase is the one that is in charge of converting glycogen to glucose 1-phosphate, and we're going to study those reactions in detail. That glucose 1-phosphate, as you can see, leads to the conversion of glucose. Then, when we are talking about how is glucose converted into glycogen, then glycogen synthase will be the enzyme that is going to be catalyzing that reaction. So, phosphorylase breaks down glycogen, while synthase is going to synthesize glycogen. And as you can see, those enzymes are also present in skeletal muscle. Now we are going to look in detail specifically at the breakdown of glycogen in the cell. So the step 1 in this reaction is going to be the release of glucose 1-phosphate from glycogen. So as you can see, this molecule that says glucose N represents the polymer of glucose. And remember that that is what glycogen is. Then, the enzyme that catalyzes this reaction, as I mentioned before, is known as glycogen phosphorylase. And you will hear me either say glycogen phosphorylase or just simply phosphorylase. So, glycogen phosphorylase is going to take that glycogen molecule, and the glycogen molecule is going to undergo phosphorylasis, which means that we are going to be removing a glucose subunit with the use of phosphate. The result of that is that now our glycogen has one less glucose molecule, and the product of that reaction is going to be glucose 1-phosphate. On step 2, in the breakdown of glycogen, as you can see, we're going to be remodeling that glycogen substrate to permit further degradation. So step 1, which is the use of the phosphorylate, as you can see, is going to happen to about 8 glucose molecules. Remember that this is just representing specifically just a segment of glycogen. So as you can see, we're going to have 1, 2, 3, 4, 5, 6, 7, 8 glucose being removed specifically from a specific branch, okay, in core of our glycogen molecule. After we have produced those 8 glucose 1 phosphates, understand that then a transferase enzyme is going to take the last remaining 3 glucose molecules and is going to transfer it to the core segment of the glycogen. Okay, so once that happens, then we're going to have a particular enzyme that is called alpha 1,6 glucosidase, and that is what's going to be removing the glucose molecule that it is the first glucose residue in the branch. Because remember, that glucose molecule is going to be bonded to the core segment of glycogen through an alpha 1,6 bond. So because of that, we need a specific enzyme to cleave it. Remember, phosphorylase is in charge of hydrolyzing the alpha 1,4 linkages within your glycogen molecule. On step 3, as you can see, we need to convert that glucose 1 phosphate into a molecule that can be utilized through the process of glycolysis. So an enzyme that is called phosphogluconeogenesis is going to catalyze the conversion of glucose 1 phosphate to glucose 6 phosphate. And the type of enzyme that phosphogluconeogenesis is an isomerase. Because when we compare the structures of glucose 1 phosphate and glucose 6 phosphates, they're going to have the same chemical formulas. It's just that our phosphate group was moved from position 1 to position 6. Understand that glycogen utilization is going to look differently on the different organs that store specifically glycogen. When we look at glycogen utilization in the liver, understand that liver is going to mobilize glucose from its glycogen storage via the phosphorylate reaction that we just discussed. Remember, after the phosphorylase has created that glucose 1 phosphate in essence, then the phosphogluconeogenesis is going to be creating that glucose 6 phosphate. Now, understand that that glucose 6 phosphate, which is at this point in the reaction, then is going to be transported. I'm just going to highlight the steps. Is then transported, okay, into the endoplasmic reticulum, and then the glucose 6 phosphatase is going to convert glucose 6 phosphate down to just glucose. Then the glucose molecule is going to exit the endoplasmic reticulum, and as you can see, that glucose molecule can now be released, okay, into the cytosol, and then after that, it's going to end up in the bloodstream. One of the interesting things that happens in the liver is that some of that glucose, it actually remains inside the liver because there's an enzyme that is called glucokinase. And glucokinase is going to be converting glucose to glucose 6 phosphate. But as you can see, that once you have glucose 6 phosphate in the liver, is going to go back into endoplasmic reticulum, because one of the main things is that the glycogen that is stored in the liver is the source of having glucose in the bloodstream. So ultimately, even though some of this glucose that it is generated is converted back to glucose 6 phosphate, it actually ends up coming back of like glucose to be released into the bloodstream. Now, when it comes to muscle glycogen utilization, we have a little bit of a different pathway, because as you can see, Muscle glycogen serves primarily the energy needs of muscle tissue itself. Now, remember, during prolonged physical exercise, what is going to be happening is that we're going to have that glycogen, okay, and then the phosphorylase enzyme is going to convert it to glucose 1 phosphate, then the phosphoglucomutase is going to convert it into glucose 6 phosphate. Then our glucose 6 phosphate, as you can see, has two routes. It could either be on route number 1, it could be taken to the ER in muscle and be dephosphorylated through glucose 6 phosphatase to make just glucose, okay? But understand that when that glucose is released back into the muscle tissue, one of the things that exist inside muscle is hexokinase. And if you remember glycolysis, hexokinase is the first enzyme that is utilized in the glycolysis pathway in order to retain glucose through glycolysis. So, since hexokinase has a higher affinity to glucose compared to glucokinase, then what happens is that glucose 6 phosphate can be diverted into another pathway, which is glycolysis, where we're now going to be taking that glucose 6 and converting it into pyruvate. Remember, when we have pyruvate around, in the case that we do not have oxygen, then that pyruvate can be converted into lactate, okay? And we're going to see how that lactate that is produced in the muscle, then it can be released from the muscle and be converted back into glucose through other pathways. But understand that the major difference between the utilization of glycogen in liver and in muscle is going to be the conversion of that glucose 6 phosphate after the glucose molecule has been released from the ER. In the case of the muscle, we can convert it to glucose 6 through a hexokinase, and in the case of the liver, we have a glucokinase that will be converting it back to glucose 6 phosphate.